Thanks so much, everybody, for coming. It's a real pleasure to introduce as our seminar speaker today, Brian He. So I'd also very much like to thank Thermo Fisher for sponsoring his visit today. So Brian completed his bachelor's degree at Stanford in computer science with a minor in English literature. And something I actually learned just a couple of minutes ago was that in figuring out what PhD programs he might like to apply for, he applied to both computer science and English PhD programs. And so I think that's just very interesting because I think one way or another you wanted to work on language is what I'm taking away. So he ended up picking computer science, and so he did his PhD in EECS at MIT. And so I first became aware of his work in a paper from his PhD that was learning evolution and escape of viral mutations, which I think was really one of the first papers to use kind of modern ML for natural language processing for, I think it was grammar kind of specifically on biological sequences and doing this in an impactful way. And this is something that, we're now just kind of surrounded by in the space of ML for proteins and for biology. And so following his PhD, Brian was a Stanford Science Fellow at Stanford University, while also being a visiting researcher at Meta AI. And so I just think this is really, really interesting, and I very much encourage trainees out there. So Brian found this way, both to be in Peter Kim's lab at Stanford while also being a visiting researcher at Meta. We talk a lot about wanting to, find interdisciplinary spaces and interdisciplinary training, but one way to do that can just literally be to spend some of your time in Peter Kim's lab and some of your time at Meta. And so there's been a number of impactful papers that have come out from in addition to Brian's work himself, also just from the EvoScale team at Meta, and Brian has been a first author on all of those papers that I'm aware of. So it's just phenomenal to me to see the impact he's had in a very short amount of time in this space. So Brian started his lab at Stanford in chemical engineering and data science in the fall of 2023, where he's also an investigator at the ARC Institute. And since then, his team has really continues to wow the field with their innovations and showing what is possible using principles from language modeling in biology. And so I'm going to not steal his thunder, and without further ado, please join me in welcoming Brian He.
Thank you, Hannah. That was a really thorough and wonderful introduction. Very kind. Yeah, so I'm excited to be here. The broad theme of my research lab is learning patterns across evolution to enable design from the molecular to genome scale. And I wanted to start with this quote. Actually, this is from Frances Arnold's Nobel lecture, where she says, today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it. And so the idea is there's been tremendous advances in technologies for genome sequencing and genome synthesis and assembly and even genome editing, but it's still very hard to generate completely new sequences of DNA, especially if these sequences are very different from nature. And fundamentally, one of the reasons why it's hard to write and read and understand things in the language of DNA is fundamentally this is a foreign language to us as humans. And so, for example, here is a cartoon of what's often referred to as the central dogma of molecular biology, where information typically flows from DNA to RNA to protein. But at each step in this information flow, the information itself is encoded in a completely unintuitive language. And I probably look at protein and DNA sequences almost on a daily basis, and I still probably couldn't tell the difference between a completely random string of like ACGT and the DNA that comes from an actual organism. But there is information in these sequences. It's not just white noise. So how do we actually extract this information from raw sequences? So over the past few years, there have been really tremendous advances in computational tools, especially from the machine learning community, in learning higher level information from raw sequence information. And fundamentally, these models are trained with very simple objectives like predicting the next token in a sequence of tokens, where a token could be an English word, for example, or a token could be a DNA base pair. And I think all of us could probably predict the next token in that first sentence. So to be or not to, if anyone has read Shakespeare, they could complete this with be. But GTG, CAT, CT, I don't think any of us could predict the next base pair in the sequence. But perhaps to a model, these two tasks are equivalent in intuitive difficulty or something like that. And a key idea in this field called language modeling is that even though next token prediction seems like a very simple task, actually, if you train a model to do next word prediction in language on a sufficiently large and complicated data set, then actually next word prediction encompasses many complex tasks within it. So for example, in this first sentence, in my free time, I like to code could be a valid completion of this sentence, but in my free time, I like to banana would not be. The idea there is you expect a verb in this position. So a model that's doing good next token prediction in language has to at least have some understanding or simulate an understanding of grammar, but it's not just grammar, right? So in the second sentence, I went to the store to buy papaya, dragon fruit, and durian could be a valid completion because it's a fruit that you can buy at a store. But even though squirrel is a noun, you probably couldn't buy a squirrel at a store, for example. But also, other things are encoded in next word prediction too, like facts about the world, or how words are associated with human sentiment, or translation between languages, or spatial reasoning, or even math. All of these complex tasks are potentially encoded in sequences, say, from the internet. And the idea is that even though large language models are not learning an explicit world model, the world has left enough of an imprint on text from the internet so that the model can learn facts from the world. So what about in biology? So in biology, we sometimes like to think of genotype as the DNA sequence that encodes for complex phenotypes via some biological mechanism. And it's often these mechanisms and these phenotypes we want to understand in biology. But also, it's beyond certain phenotypes and certain, say, high throughput methods, it's often very difficult to collect data, especially sufficient data to train large machine learning models at the level of mechanism or phenotype. But thankfully, actually, it's been very easy to collect data at the level of DNA sequences through advances in, say, next generation sequencing. So a key idea in this work and a key hypothesis is that even though we can, for example, train models on DNA sequence, and even though evolution acts on phenotypes, the effects of the selection get written back into the DNA sequence through, say, evolutionary selection. And in the same way that the world has left an imprint on sequences from the internet, the world has also left an imprint on sequences from natural organisms through evolution. So this is a key hypothesis. This hypothesis, which is an intriguing one, would be that even if we train a model on a very simple objective, like predicting the next base pair in a sequence, if you train on a sufficiently complex data set, then this next base pair prediction task would teach you things about which mutations are correlated with health or disease, or how DNA bases form codons and how these codons encode for amino acids and how these amino acids come together to make protein molecules, or how RNA bases pair with each other, or even how genes clustered together into bacterial operons would tell you perhaps something about systems biology. So this is a very intriguing hypothesis, but is this actually the case in practice? And Hannah alluded to this in her introduction, but a lot of the previous work and current work in the community has been training models on not sequences of DNA, but sequences of proteins. And here you have a vocabulary of 20, say, canonical amino acids. And you train models that can predict the next amino acid in the sequence, or a related task where you hide certain amino acids from the model and ask the model to fill in the blanks using the observed amino acids in the context. This is similar to games like Wheel of Fortune or Hangman or something, if you're familiar with that. And you train these models on hundreds of millions of protein sequences. And the idea is that through evolution, the model learns how evolution has put amino acids together to form valid protein sequences. And suffice to say that these protein language models have made a big impact in protein biology. And more generally, models like this that are trained on large evolutionary databases have really revolutionized the study of molecules. So for example, methods for de novo protein design by David Baker and methods for protein structure prediction like AlphaFold were awarded the Nobel Prize in chemistry last year. And this was very well deserved. Molecules are still are very complicated. We are still very interested in molecules, but we're also interested in how these molecules come together to, say, make a complete organism. But even a very simple single-celled organism is orders of magnitude more complicated than any individual molecule. And we not only want to understand things at this level of complexity, but we also potentially want to be able to redesign or control or engineer cell state and things at the whole cell level. How do we even do this with current methods? And actually, the current methods probably can't get us there. But today, I wanted to lay out, say, a roadmap towards whole genome design. And this talk will talk a lot about computational methods and algorithms that often have a lot of jargon. So I'll try to give intuitive explanations of what all of these things are. But roughly, we will start with this class of methods called unsupervised pre-training. And the idea behind this class of methods is who would you rather train? Here's an intuitive explanation, perhaps. And say you want to train someone who is new to science and you want to train them to pipette. So would you rather train a high school student to pipette, or would you rather train a baby? And I think most of us, even though the baby and the high school student start off not knowing how to pipette, the high school student has still learned kind of generalist knowledge. The high school student can communicate in language and has fine motor control, all these things that the baby doesn't have. So one idea in machine learning is you often want to start off, especially for complex tasks, with a model that just has generalist understanding of, say, the basic rules of biology, like what a start and stop codon are and the genetic code and things like this. Cool. So what do we actually use to pre-train the model to get this generalist information? Well, over the past 20 or 30 years, people have actually been sequencing the complete genomes of millions of organisms and have been depositing these genome sequences in publicly available databases. And in theory, embedded in these DNA sequence data sets is all the information, or is the information required to create whole organisms. So we can train language models on these DNA databases. So this brings us to a model, a DNA language model that we put out last year. This is a model we called EVO, or EVO1 for the purposes of this talk. All this model is doing, it's actually a very simple task that this model is taught to perform. All this model is doing is it takes in a sequence of DNA base pairs and it tries to predict the likelihood of the next base pair in the sequence. That's all the model is doing. The model, though, is fairly large. It's a 7 billion parameter neural network. It's trained on a very large database. This database in EVO1 consisted of prokaryotic organisms, so like archaea and bacteria, some bacteriophage. And the idea is that these genomes are simple and would give us a good idea of whether this language modeling paradigm would even work on DNA. There's one idea that's important to the EVO1 model that's also important to communicate here. It's this idea that even though we're only training a model on DNA sequence, because of the beauty of the central dogma, this DNA sequence directly encodes for RNA sequence, and that RNA sequence directly encodes for proteins. And in prokaryotic genomes in particular, those sequences are also contiguous. There's no splicing in the coding sequences in prokaryotes. And so even if we're only training a model on DNA, we could potentially also get protein language modeling and RNA language modeling and regulatory DNA language modeling for free out of a DNA genome foundation model. But another important idea behind EVO1 is that we can understand not only a single gene or a single protein coding sequence, but we can also place that gene in the context of, say, multi-gene systems, or even, in some cases, complete genomes. So we actually teach, we train EVO1 to be able to process an input sequence of 131,000 base pairs. So at that scale, you can maybe fit simple phage genomes and things like that fully within the context of the model. So we trained EVO1 only on prokaryotic genomes. This left out a third domain of life, the eukaryotes. And this is an important domain because it includes humans. The problem with eukaryotic genomes is that they're much longer and much more complex than prokaryotic genomes. So we needed to do a lot of work in data curation, making sure that we were not training the model on repetitive sequences or telomeres or centromeres or things like that, and a lot of work in actually scaling the model to process longer sequences. So this led us to train a model that we're calling EVO2. We put this model out as a preprint earlier this year, and this is a model trained on all domains of life. Again, simply doing next base pair prediction on everything from a very simple bacteria to the genomes of plants and animals, and one copy of the human reference genome in this case. EVO2 now also has 1 million base pairs in its context. So we can still have molecular level resolution, but we can place these genes in the context of, say, a complete minimal bacterial genome. We also scaled the model to 40 billion parameters with the help of NVIDIA, who graciously supported us with 2,000 H100 GPUs to train this model for around 2.5 months. And we were able to scale these models to 10 to the 24 floating point operations. We also make this model fully open. So it's not only are the model weights publicly available, but our code for training the model and running the model and the training data are all open sourced. And so now I'm going to proceed into the main thing I wanted to talk about today, which is how do you actually use these models to generate new sequences. So these models are generative models in the same way that large language models of text or images are generative models, and you can sample from them to have the model write code for you or generate a new image for you. In the same way, we can also sample from these models to generate DNA sequences for us. And the way this works is basically we use the fact that the model can predict the value of the next base pair. So say the model samples an A in the first position, we can use this A to predict that maybe T is in the second position. We can then use AT to predict that T is in the third position. And basically, one base pair at a time, predict the value of the next base pair in the sequence. And in this way, these models are generative models. What we did with the original EVO models is we tried to use these out of the box for generation. And for example, with EVO1, we did this kind of what's called autoregressive sampling, but we sampled 1 million base pairs. And we thought that these sequences would be completely nonsensical and complete garbage. But actually, when we started analyzing these sequences from the model, along certain bioinformatic, with certain bioinformatic analyses, these genomes look somewhat reasonable. They have the right predicted coding density, for example. There's some predicted secondary structure in the proteins. We actually find tRNA sequences on these genomes that cover all 20 canonical amino acids. So in some sense, these are maybe, we use this analogy, like the first versions of image generation models would generate blurry images, and they've since gotten a lot better. This first version of the EVO model was generating, say, like blurry versions of genomes. We actually tried this with EVO2, which is a model that has been trained on a lot more data and with much larger scale. And we actually see that the genomes look less blurry. So maybe with EVO2, there's more genes on the genome that map to things that we know and understand in nature via like a simple BLAST query. But also these genomes are still missing many essential genes. There's lots of aspects that make us think that these genomes are not going to work if we actually put them into an actual organism. So there's still a lot of work to do to improve the the generation capability of the model. So this brings us to the second technique that we've been using to improve these models' ability to generate new DNA sequences. And this is something called supervised fine-tuning. And intuitively, you can just think about this like the actual specialization task. Like if you want to teach someone to pipette, you need to put in the 10,000 hours and just perform the task over and over again. So in this case, with the original EVO1 model, we did this supervised fine-tuning task. And the goal of this generative task is to design a complete CRISPR-Cas system. So these are systems that are often used for genome editing. They're actually pretty complicated. They involve a fairly large protein. Cas9 in this case is more than 1,300 amino acids. And it's bound to a guide RNA, which is a non-coding RNA, which itself is composed of two different non-coding RNAs, a tracer RNA and a CRISPR RNA. So as part of the fine-tuning task, we specialized EVO1 in this case by training it on 70,000 examples of CRISPR-Cas sequences from nature. And we also prepend a special token so that if it sees this token, the model knows to generate a CRISPR-Cas operon directly after. We first verified that it's actually important to start off with a generalist model rather than training a model from scratch. So on the top is what happens if we sample from a model that was only trained on CRISPR-Cas but was not pre-trained on all the prokaryotic organisms. We can't really successfully generate any Cas9 proteins at all. But if we start off with a model that has been pre-trained, then this model also and then fine-tune that model on CRISPR-Cas systems, then that model can actually generate pretty coherent CRISPR-Cas sequences. So here's one example of a CRISPR-Cas operon that we designed. In this case, this is an operon we're calling EVO-Cas9-1. It has a full-length Cas9, and we can detect tracer RNAs and CRISPR RNAs on the sequence. The AlphaFold 3 structure prediction resembles that of known CRISPR-Cas systems. We can then chemically synthesize this DNA and test it in the lab for in vitro cleavage of double-stranded DNA. And actually, this designed CRISPR-Cas system has comparable in vitro cleavage to SP-Cas9, which is this state-of-the-art natural system for genome editing that's often used for genome editing. But importantly, our designed CRISPR-Cas sequences have less than 80% sequence identity to any Cas9 we can find in nature. For context, this is enough to break any patents around CRISPR-Cas systems. So at least in the eyes of the patent office, these molecules are novel. We can also do the same thing. I won't spend too much time on this, but we can also fine-tune the model to generate these protein DNA systems, these transposable elements that are composed of a protein that forms a homodimer that interacts with two flanking DNA hairpins. And we can fine-tune the model to generate examples of these transposons and verify, again, in vitro that these transposable elements are capable of both excision and insertion into genomic loci. So there are some limitations of supervised fine-tuning. It requires lots of data to specialize the model. In this case, we have tens of thousands of sequences from these natural systems. Also, if you fine-tune a model on a data distribution and then sample from this distribution, you're often maybe prone to memorization. So we wanted to then see if we can also prompt the model without doing fine-tuning to have it generate interesting things. And for this, we use a class of techniques often referred to as prompt engineering or context engineering or sequence conditioning. One intuitive way I like to think about it is, say you have two different prompts that you give to a student. So one prompt is provide the balanced chemical equation for oxygenic photosynthesis, then name the two major stages of this process, specifying the primary inputs and energetic outputs for each stage and their precise locations within the chloroplast. Maybe some people here could actually answer this question. Then you can compare this prompt to this question: what if humans could photosynthesize? And with one prompt, you might elicit a lot of memorization and recall from the student. With the other prompt, you might actually elicit a lot of diversity in the responses and creativity from the student. So we wanted to see if we could actually prompt Evo to be creative in its generations. And for this, we actually leveraged the property that the model learns information across multiple genes. So you can often prompt the model with a gene sequence from nature and then have it generate another gene in the response. And in prokaryotic genomes, functionally related genes are often directly next to each other on the genome sequence. So we can prompt the model with conserved regions of the genome and actually get it to recall some parts of its training data. So in this case, this is a highly conserved operon. We can prompt with mod A, get things that look like mod B in its response. We can prompt with mod B, get mod C in the response. But then we did this interesting experiment where we asked, what happens if we now prompt the model to generate a hypervariable part of the genome? In this case, we prompted the model to generate these genes. They're anti-CRISPR proteins. Anti-CRISPRs are very diverse because they're involved in the bacteriophage arms race. And many anti-CRISPRs will inhibit CRISPR, for example, by binding to Cas9, for example. So here, anti-CRISPRs are very diverse. A lot of them are actually hypothesized to emerge via de novo gene birth from like intergenic DNA sometimes. So we wanted to see what happens if we prompted the model with anti-CRISPR associated proteins and then generate this hypervariable region of the genome. And what we find actually is that we do find genes in the generated sequence, and these genes have anti-CRISPR activity. So in this case, we show that these genes can inhibit SpCas9. But then for some of these anti-CRISPR proteins, we can then look at all the proteins and genes that we've ever discovered from nature. And in some cases, these genes have no significant sequence identity to anything in nature. So these genes are for all intents and purposes de novo genes. And I think this is pretty compelling evidence that the model is able to generalize non-trivially in sequence space while maintaining function. And actually, we do this analysis that shows that basically, in order to recombine sequences from nature to reconstruct the model-generated protein sequences, the patterns for the Evo-generated anti-CRISPRs are more consistent with these de novo designed proteins from like David Baker's group than by like trivial recombinations of existing proteins. We then do this across many putative functions. So we actually then generated 100 billion base pairs of AI-generated genomic sequence and deposited them all on the internet. Cool. So the last kind of technique that I wanted to talk about is something called inference time guidance, where we pair a generative model with another model that guides its generations. You can intuitively think of this as the generative model like Evo2 being a proposal, and then you have another critic or a scoring function that can accept or reject the outputs of the model and maybe even guide its generations towards more desirable outputs. So for here, we wanted to do this design task, which is a little bit of a contrived task, but we wanted to make it very artificial. So here we're using Evo2 in this case to generate de novo sequences of mammalian genomes where we can control the regions of the genome that are more or less accessible in the chromatin structure. So lots of mammalian genomes will modify the chromatin architecture to make the DNA more accessible to proteins to bind. And so we wanted to design this with Evo2. Evo2 does not have any explicit understanding of chromatin accessibility. So we then pair the model with these models that do predict chromatin accessibility from sequence. These are models Informer and Borzoi from DeepMind and Calico. So these models take in DNA sequence and predict chromatin accessibility. And our design task, it's a little bit contrived, is we wanted to encode Morse code into the chromatin architecture, where a narrow peak is a dot, a wide peak is a dash, and the heterochromatin are the spaces. To do this, we sample autoregressively from Evo2 with, I think, I did this. I think it's like 32 base pairs at a time or something like that. And then after the model generates these 32 base pair chunks, we'll accept the generations that have the desired chromatin architecture, reject ones that don't, and then the ones that we accept, we continue generating off of those. So it's a simple beam search algorithm, if you're familiar with that. And in this way, we have a way of basically steering the model to generate chromatin architecture that we want. And we don't have to generate all 20,000 base pairs before scoring it. We generate a chunk at a time and can accept and reject intermediate outputs of the generation. So here is a GIF that shows one design trajectory. On the x-axis is the position along the genome sequence. On the y-axis is the predicted chromatin accessibility. The bold yellow line is the design that we actually want to achieve with the model. So I'll leave it as an exercise for the reader what Morse code message we encoded here. But a narrow peak is a dot, a wide peak is a dash. And then this squiggly white line is the actual predicted chromatin accessibility of our designed DNA sequence. And each frame in the GIF is a new chunk that gets sampled by the model and then accepted as having the right chromatin architecture. And you can see basically the model generating from left to right a DNA sequence with the desired chromatin accessibility. We can actually test these designs in the lab. So for this, we partnered with a company called Anza Biotech that specializes in long DNA synthesis and assembly. So they helped us assemble 20 KB sequences. We can then insert them into mouse ES cells and then verify chromatin accessibility with this assay called ATAC-seq. And actually, the design results match pretty well the predicted chromatin architecture from the model. So here, the model is actually able, and we're actually able to kind of write Morse code messages in the chromatin architecture on this de novo DNA sequence. And so the message that we encoded was Evo2, if you didn't get that. So that's the message that we encoded. And this shows that we can actually control the chromatin architecture in mammalian cells with a generative model. More broadly, we think this general paradigm of coupling a very capable generative model with a critic or a reward function or a scoring function that predicts a function of interest is a very powerful paradigm, not just for generating de novo sequences with desired chromatin architecture, but maybe even designing sequences with desired protein structures. Or if you have a model that can predict any function of interest from a sequence, you can then couple it with a generative model of sequence and then guide that model to generate things with your whatever function of interest that you want. Right. So in the final part of the talk, I want to talk about a recent story that we pre-printed a couple weeks ago. And here, we're actually going to put all these computational techniques together to achieve generative design of complete genomes, in this case, the genomes of bacteriophages. So suffice to say that designing end-to-end a complete genome sequence is a very complicated task. So at the genome scale, you're not just designing one or two genes, you're designing multiple genes. In bacteriophage genomes, these genes are sometimes overlapping with each other. These genes also interact with host proteins that are not on the genome sequence. And you're not just designing genes, you're designing all the non-coding sequence in the genome, and all those things need to be in the right stoichiometric stoichiometric proportions for the phage to work and things like this. So it's a very complicated task to design these genome scale sequences. We picked phages because their genomes are pretty short, though. So in the tens of, or the low kilobases. Phages are also an emerging therapeutic modality for multi-drug resistant bacteria. So here is a very famous case from a couple years ago. This man, Tom Patterson, almost died from a multi-drug resistant bacterial infection in his lungs, but his life was saved by a phage. And so here he made actually a full recovery, and his wife is holding a photo of the phage that had saved his life. So for our bacteriophage design task, our goal is to design bacteriophages that use as a design template this phage called PHYX174. It's actually a quite complicated, it has a quite complicated genetic architecture. The PHYX174 genome is 5.3 kilobases. It has 11 genes. Some of the genes actually overlap on the actual sequence. So the same DNA sequence will encode for two different proteins. They're just out of frame with each other. It also has several regulatory elements. It's a very complicated genome to design. So how do we actually achieve this very complicated design task? Well, we can use all of those tools in our toolkit for improving generative design of biological systems. So we, in this case, start off with pre-trained models. We, in this case, start off with Evo1 and Evo2 as our pre-trained models. We then do supervised fine-tuning on these models. So we actually take 15,000 examples of phages from the same family as Phi-X174. It's a phage family called Microviridae, and we specialize the model to predict the next token, but only on the sequences of these Microviridae genomes. We also include these special tokens, again, so that if the model sees this special token when you're using the model, it knows to generate a phage genome in its output. And then we do this prompt engineering. So we actually had to do a lot of playing around with the parameters when we were sampling these genomes. So, and this is one result that I thought was pretty interesting. So we prompted the model not just with the first few tokens, the special tokens that we introduced during fine tuning, we also prompted the model with the first few sequences of the Phi-X174 genome as it appears in the actual training data. And it's a circular genome, but in the computer, the genome does have to start from somewhere. So we prompted with that. And we found that if our prompt from Phi-X174 was too short, the model would not have sufficient conditioning. It would just generate random phage genomes that looked nothing like Phi-X174. So we didn't provide the model with enough information about Phi-X to tell it to generate Phi-X. But if the prompt was too long, then the model would just regurgitate Phi-X174 from its training data and just give us the same sequence of Phi-X. And in the most extreme case, right, if you prompt with like half of Phi-X174, half of that sequence is guaranteed to be identical to Phi-X. But we actually found this sweet spot in the length of the prompt, that balance between giving us phage genomes that resembled Phi-X174, but that were still different in interesting ways, and ways that we could maybe experimentally determine if these are like novel, viable genomes. So we have this sweet spot for EVO1, it's around 4 nucleotides. For EVO2, it's around, I think this is an old slide. It's around 20 nucleotides for EVO2. The final step in the process then is we then apply this inference time sampling. So we have the model actually propose a lot of different Phi-X sequences, and then we accept sequences that have a genetic architecture that resembles that of Phi-X174. One interesting detail is we wanted to see that the generated genomes had predicted ORFs that generally match the ORF structure of Phi-X, but many off-the-shelf methods for predicting ORFs didn't work for Phi-X, so we had to write our own ORF caller. This is related to the ORFs sometimes overlapping in the sequence. That is not great for the publicly available ORF callers. We also wanted to test these genomes against a very specific host. So in this case, we also constrained the spike protein to be similar to that of Phi-X174. So this gave us a better shot of these phages actually being able to infect the host that we're actually screening against. Okay, so we do all of these things and we get a set of Phi-X174-like viral sequences that we can then experimentally test in the lab. And here we basically have a very medium throughput system where we can just order the DNA as double-stranded DNA fragments and then turn them into circular DNA via just a simple Gibson assembly reaction, and then transform this DNA into E. coli C. And basically, if the E. coli dies, then the phage is probably working. But if the E. coli is able to grow, then the phage is probably not viable. So this let us screen around 300 phage designs. And we found that 16 of these AI-generated phages actually had pretty robust viability. So these are completely synthetic phages generated by an AI system. Here's a phylogenetic tree in the middle of the slide that shows where these phages would fit in relation to natural phage genomes. And interestingly, one of the generated phages has less than 95% average nucleotide identity to any known phage in nature. And there's this committee, the ICTV, that has set this threshold where anything less than 95% ANI technically counts as a new species. So in one sense, the model was able to generate a new phage genome that if we had isolated it from nature would be considered a new species of phage. there's also interesting new structural changes to the phage. So here is one phage that we called Evo-Phi36, and we actually determined a 2.9 angstrom resolution cryo-EM structure of this phage. And the reason why we were interested in the structure of this phage is that the DNA packaging protein in Evo-Phi36 is actually a packaging protein from a very distant, an evolutionarily distant phage, this phage called G4. And people have actually previously shown that if you just put the G4 DNA packaging protein and swap it with Phi-X's DNA packaging protein, that phage is not viable. But the AI system was able to make this gene J swap and mutate enough of the genetic background to make this phage now viable. The DNA packaging protein, though, is very short. It's missing a number of amino acids in the middle of the protein. So we wanted to understand its structural consequences. It's actually amazing that this phage even works. The AlphaFold structure is also not correct. So it's good that we solved this with cryo-EM. And we can actually show that some of the residues in the latter part of the, or in the, yeah, we can show that some of the N-terminal residues of the protein are not resolved, potentially indicating like a flexible region of this protein. And we've since like done additional structural work. We can even resolve some DNA in this. It's pretty cool. But I think it's a pretty cool story. This is an example of like a structural and mechanistic characterization of an AI-generated genome, actually. So people might say that AI is going to take all the jobs and make biochemists and structural biologists useless. Actually, I think maybe there's a future where a lot of biophysical and biochemical and structural characterization could be done actually on AI-generated genomes. And so the structural biologists are definitely not out of a job with AI. In fact, AI might make the job a little like even more fun and have things like this. Cool. Okay. Other interesting things about these phages, some of them do seem to have higher fitness than the wild type Phi-X genome. So for example, we do this competition assay and we sequence at multiple time points and show that a few of these phages reliably across many different, 3 different competitions will reliably outcompete the wild type Phi-X genome. And another interesting thing we see is if we just pool all of these AI generated phages into a phage cocktail, they much more rapidly overcome bacterial resistance on the side of the host. So we actually have these 3 resistant strains. They're resistant to the native Phi-X genome, but after just 1 to 5 passages with the AI-generated cocktail, we're able to actually overcome the resistant strains, whereas passaging native Phi-X for the same number of passages does not overcome resistance. Cool. So this was an example of us being able to put all these different algorithmic, I guess, parts together into a single pipeline. So generating phages required pre-training models on just general DNA sequence from nature, but also specializing the models on microviridae, the right sampling and like creative actually use of the models at inference time, and then also scoring the models and accepting and rejecting models that met various rationally designed criteria. Right. So I wanted to close by recalling this quote by Frances Arnold, where at the end of this quote, she does say that evolution knows how to cut and paste and modify sequences from nature. And so how I see this work fitting into the broader, say, arc of biology is that there have been technologies for reading, writing, and editing DNA. And now by learning from evolution, we also have technologies for composing new DNA sequences. And together, this can actually enable genome scale design. And Phi-X 174 was actually the first complete DNA genome ever sequenced by Sanger in 1977. It was the first genome also chemically synthesized by Craig Venter in 2003. So these are the examples of DNA read and DNA write. And now we see this as like the first example of DNA compose or DNA design at the whole genome scale. Great. So in summary, hopefully I've convinced you that we're making a lot of progress in our ability to generate new biology from the scale of single genes to genomes. So for example, we can design de novo anti-CRISPR proteins. We can design multi-gene systems like CRISPR-Cas that involve protein RNA interactions, or these transposons that involve protein DNA interactions. We can also design multi-kilobase sequences of mammalian genomes to have programmable chromatin architecture. And we can design complete genomes at the scale of, say, a complete bacteriophage. So I glossed over a lot of information in this talk, but we do make all of the code of these models publicly available. We make all for EVO2, we make the training code and the inference code, the training data publicly available. So the links to all of that are on this slide. And these are references to the papers in case you're interested in learning more about the different things that I talked about today. Finally, this is work done. So EVO itself is a major collaborative effort across many different teams spanning people with machine learning engineering and infrastructure and architecture development, people in computational biology working on data and evals, people working on interpreting the model, people working on experimental biologists who actually make the DNA and test it in the lab. This is a collaboration not only at Stanford, but also with Arc Institute and NVIDIA to actually train the EVO models. And the work on using EVO to design new biology is led by my group. This is a photo of us on a recent trip to Korea, actually. And so finally, thank you all for your attention. With the remaining time, I'm happy to take any questions.
Thank you so much, Brian. That was fantastic. Are there questions for Brian? Do you want to shout?
Generate these phage genomes. The success rate maybe like 5%. I wonder, can you? To me, it's like.
Yeah, and with the success rate, we actually think we have a lot of designs that we like really wanted to work, but they were like crazy, and we wanted to just see if those are working. So if we subset to the more conservative designs, maybe our success rate is actually much higher than 5%. But yeah, exactly. Like now that we have around 280 sequences we are able to successfully synthesize and test, we can retrospectively see where along which metrics actually correlate the most with design success. We can either fine-tune models directly, or it's hard to fine-tune models on 300 data points. So maybe we can just train like a simple classifier or something on that data, and that can help us boost our success rate if you just care about hit rate.
Yeah, really interesting talk. I wonder if in your competition experiment, I'm assuming you did it at low multiplicity, so each virus gets its own host cell. And I wonder if you've tried it at high multiplicity where the viruses can be competing for the same resources in the cell, or maybe would speculate on what would happen then.
Yeah, that's true. Like we like titer all the virus to make sure we're trying to infect with the same MOI at the beginning. There's a lot you could do with this. Like bumping up the MOI, like that could be cool. We also see actually, this is interesting. This is an interesting point. We also characterize the phages for how fast they're able to perform lysis, and we see from this that like our competition results are not 100% correlated with lysis kinetics, but maybe under like other kinds of conditions like higher MOI or something, like the lysis kinetics become a lot more important. So yeah, that's a cool idea.
Yeah, just building off the last two questions. I was wondering if you've come up with principles on how to design the experiments to get kind of higher success rates on predicting ones that do better. So like there's been quite a bit of model development, I think, at least in some of the chemical engineering spaces, where you can predict designs that fail, and that's pretty robust depending on your training set, but it's harder to sample the ones that increase fitness. Are there ways in which you've come up with kind of efficient ways to sample the kind of positive fitness landscape rather than try to predict things that kind of fall down of the fitness landscape?
Yeah, that's a good question. And you're alluding to like how the naive way for increasing your success rate is like, instead of sampling 2,000 sequences and accepting like 10% of them, you sample like, you have a more stringent filter for success rate. You sample like 1 million sequences and then pick again the top, whatever, 100 or whatever. That might be inefficient if you're like not sampling from the positive examples frequently enough. So one thing people do is actually then try to retrain the model and kind of bias its generation towards high fitness, in this case, examples. And there's even special like losses you can provide to the model to where you like pair a positive example with a negative example and say like, try to increase the likelihood of the positive example and decrease the likelihood of a negative example. And it's like a paired or like a contrastive loss. These are things people do to improve just like the in silico success rate of the generative model. But yeah, and we haven't tried this, but we've done it for different protein level tasks, and it's a good way of improving the success rates.
Yeah, very nice talk. So I'm curious about the prompt engineering part. So when we try to like design a phage genome, we first need to provide a existing like phage genome to provide some conditional guidance. But as you have said, like in the slides, we don't want to like provide too long or too short these genomes to like ask the model to generate like innovative like these genomes for phage. So I was wondering if there is a standard way to give these conditional like guidance instead of like do these trials and errors. And if we want to apply this generative model to a larger genomes, are these like standard methods different from the phage? Thanks.
I think there's a lot of parameters that go into sampling from the model. And I think the consensus in the field broadly is that to get the best performance out of the model for generating new sequences, you need to do a little bit of empirical like testing or like parameter sweeps. So you do need to like play around with those parameters a little bit now, and it's kind of an art. And this is not just true for DNA models. Like all these protein models like Bindcraft all recommend that you play around with the sampling parameters a little bit before launching your full design run. In some cases, like in this case, it's not like the model won't be able to, with a suboptimal set of parameters, it's not like the model will never sample something that looks good. It might just sample it at a lower or much rarer. Like you'll need to burn a lot more compute to get the same number of like good looking designs is the only thing. For larger genomes, yeah, this will also be important.
So great talk. I sort of had two questions. The first one was, and this goes a little bit beyond what you mentioned here, but I read the preprint.
Yeah.
And the preprint had mentioned that for the host tropism filtering step, right? You tried to avoid K12, but I didn't see anything specific in the methods about how you did that. Was that just the retention to 60% for the spike protein, or is there more beyond that?
No, that's it. So we already know that, yeah, we know that the wild type phi-X does not infect K12 and only infects Coli-C. So our thought is that if you constrain the spike tropism determining protein to be close to phi-X, then it would have a similar host range.
Okay, then that leads into the second one. So this phage is quite easy to engineer to infect K12, right? It only needs a couple of mutations to do so. So are you surprised that your model did not pick up the ability to infect those in that case?
Yeah, I don't know. I don't know if we hoped that the host range would not expand. We do want to try in the future to play around with that protein more and actually like screen against like a broader panel of things. So hopefully, I don't know, hopefully the model can do it. I don't know if I was surprised. It was like what we expected. But yeah, it is interesting why yeah, I'm not sure.
There's a lot of questions, but I'm going to ask a question in the interim. I wanted to ask about the data that these models are trained on, Evo 1 and Evo 2. And I know there's people out there, you know, making things like FROs to just get more sequencing data and saying that publicly available sequencing data is like a fraction of everything.
Mhm.
Do you view this as a limitation in, you know, training to generate from these?
No, we're not data limited in this area of biology. Like actually, when we were curating this Evo 2 training data, we were like actively trying to filter out genomes because there's too many. And 9 trillion base pairs, which is the Evo 2 training data, is actually not a lot. It's like 9 terabytes of genome sequencing data. Like there's a lot more out there. So I think the people going out and sequencing deep sea vents or the environment for additional genomes, that could help with diversity. And I think maybe if we had access to those genomes, we could like filter out redundant genomes or more redundant genomes in the existing set. But I think in terms of the broader question of like, are we data limited in the setting? I actually think we're not. There's too much data, and we can extract a lot more data from even the existing genomes that we have.
I think we're almost at time. So if there's one more good question.
No judgment on quality.
Hello. I don't know if this is a good question. It's really interesting that you started with sort of phage and prokaryotes because they do have these like compact genomes or operonic genomes, but a lot of the applications for this technology will ideally be in eukaryotic tools, maybe designing stuff for like T-cell therapies and whatnot. But how do you view these tools being applied to those genomes where you're starting to deal with issues with like context window, right? Where in a prokaryotic system, you'll have genes that are very similar next to each other, where that's not always true in like eukaryotic systems, or eukaryotic genomes are just full of, you know, I'm going to do quotations, junk DNA. How do you view this, adapting this tool to those types of genomic sequences?
Yeah, I mean, I actually think there's a lot of prokaryotic biology that would be useful for human sequences. Like if you think about PCR and or CRISPR, like all these technologies are mined from prokaryotic genomes, and all our technologies for genome editing were like taken from prokaryotic systems and are now like in eukaryotic systems. So essentially all the prokaryotic stuff that the models are in can be quite useful from a molecular technology perspective. But it is also true that maybe we also want to design components of human genomes for things that are like that have like cis regulatory properties, like proximal enhancers or something like that. Like there, everything will fit within the model's 1 megabase context just fine. Yeah, if you have 2 like mammalian or 2 like human genes or something that you want to re-engineer to make like a synthetic orthogonal signaling pathway, for example, you might not be able to use the Evo 2 model out of the box, but you could, for example, and this is where a lot of our thinking has been, like if 2 genes interact with each other but are not like operonized or not directly next to each other in genome, how could you adapt the technology to be useful in these settings? And it would probably look something, you know, like having the model still be able to propose new sequences or diversify sequences, but you need to couple it with a model that does learn explicitly that these 2 genes interact with each other. So that's how I would answer that question. It's a good question. That was a good question. Yeah.
Thank you.